you all for attending this lecture today, and especially Daniele and Stefan and other members of the organizing committee at AtlantECO to give me the opportunity to speak about one of the pillars of the AtlantECO project. And so today I'm going to talk about plastics and the plastisphere. And you see this is an artist's rendition of the theme of today's lecture. Oops. Okay, yeah. So the outline of my talk is here. What I'd like to do is sort of do a little bit of myth busting early on, because I know that the topic of plastic marine debris comes with a lot of facts and fiction. And I think that some of these myths perpetuate still in certainly the popular literature and media, but sometimes also in the scientific literature as well. Next, I'm going to talk a little bit about the plastisphere, the sort of origins and how it's evolved over time. And specifically, I want to emphasize the diversity, function, and fate, what we know so far about some of the key highlights of the plastisphere. And then I want to segue into the topics of degradation and biodegradation, because I know that these are areas of interest for AtlantECO and also in my own research. And then finally, I want to leave everyone with some conclusions and outlook. So first, fact versus fiction. Now for this, I need to acknowledge Eric Van Sevilla, who spoke and gave a wonderful lecture yesterday, because these 7 myths originally come from one of his outreach activities. When I normally talk about plastic marine debris, I tend to talk about maybe 2 or 3, but he has actually discovered 7 of them. And I think that they're quite relevant, and I want to share those with you and my perspectives on them. So first, there are whole islands of plastic floating around in the ocean. And the perception you see on the right-hand side is that there's these large masses of macroplastic that are floating in the ocean gyres in particular. But this photo and many photos like this appear on the web. And what they are typically are areas in coastal regions, particularly places like Southeast Asia, but other coastal areas as well, where you do in fact see large accumulations of macroplastic. But in fact, if you go to the garbage patches, which Eric spoke of yesterday, in reality, this is a snapshot of what you see on the right-hand side. And that's because when you drag a net, a manta trawl across the surface of the ocean in the Great Pacific Garbage Patch, of which this is an example, what you see is something like this. This is the result of a manta trawl, and it shows you these confetti-sized pieces of microplastic, and that's just a scale of 4 inches and what you would collect in a net tow from the Great Pacific Garbage Patch. So as a result, this has led people to speak of the Great Pacific Garbage Patch more in terms of the plastic soup. And in fact, there's an entire organization called the Plastic Soup Foundation that is dedicated to educating the public on this topic, and at some level sort of correcting this myth of plastic islands floating in the Atlantic. So this sort of piggybacks on another myth, which is plastic is everywhere in the ocean. Plastic isn't everywhere in the ocean, and hopefully that was a take-home message that Eric explained yesterday. This is from a colleague of ours, Nikolai Maximenko, doing similar kinds of modeling work, and this shows the result of what happens when you look at when particles are released, where they end up and where they concentrate. And these gyres have been confirmed with empirical data as well, but they speak to the fact that you will not encounter the similar concentrations of plastic everywhere you go in the ocean. And when I first moved my lab from Woods Hole to the Royal Netherlands Institute for Sea Research in the Netherlands, I had the opportunity to go out on our research vessel, shown in this diagram here, the Pelagia, and we did a microplastics cruise from the Azores to Sicily. And when we did that, this is an example of, again, this issue that plastic isn't everywhere equally in the ocean. And I think it's a nice sort of empirical observation of what you find when you take a manta trawl shown here, and you sample the microplastics in the floating on the surface of the ocean. You see there's some nettos where we got actually no plastic, and then sadly enough, as we ended our cruise, we got the highest concentrations we had ever encountered just off of Sicily, and we in fact weren't even able to count all the particles as a result. Okay, a second myth is, fortunately, we've already started cleaning up the plastic soup. I'm sure many of you are familiar with these kinds of efforts, perhaps the most famous of which is the huge plastic collection system of buoyant slots in the ocean cleanup. And this has been put forth as an open ocean solution to collecting plastic. But hopefully my previous slide convinces you that this is perhaps not the best way to be spending our resources in terms of cleaning up the ocean. And I think even the ocean cleanup is realizing that, because now they are intending to clean up rivers. In fact, I think they're targeting 1,000 rivers over the next 5 years. And this leads us to other examples of cleanup efforts, such as Mr. Trash Wheel, which I think is being used now in places like Baltimore and other parts of the US. And things like the Great Bubble Barrier, which is yet another solution. This is actually a picture that I took in Amsterdam. And these are so-called urban solutions to the plastic problem of cleaning up the plastic soup. So people are thinking about solutions, but anyone who works with microplastic in a scientific sense and is responsible for trying to quantify it knows that harvesting microplastics is actually really hard work. These are some of the examples of tools that we use in the business of trying to quantify microplastics, everywhere from the traditional manta trawl, where we can collect samples at the ocean surface, to sediment cores, where we can collect it at depth, and everything in between, from multi-nets that sample the midwaters, to high volume pumps, also suspended at different levels, that can collect various size fractions that aren't typically collected in manta trawls, which tend to collect upwards of everything greater than 333 microns or so, or 350 microns or so, to hydrocasts, so actually collecting water and then concentrating that water, and then subsequently looking at concentrations of plastic in the ocean that way. A third myth is that many animal species are threatened with extinction due to plastic waste. Now, this is a little tongue-in-cheek. This is actually a breaching whale, and this is a whale coming out of a canal off of Utrecht Centraal in the Netherlands. But it does sort of speak to a fear that, in fact, our wildlife will soon be replaced by artificial forms, so to speak. But when I look at this, I think, well, it's really the plankton we need to be worried about, in addition to worrying about mammals, marine mammals, and other organisms, because, of course, the plankton are at the base of the food chain. And so we need to be mindful of the fact that just because there are larger organisms that are perhaps more charismatic and people are more aware of these, we also need to make people aware of the very base of the food chain that supports the flow of carbon and energy in our ocean, and is vital to life, in fact. And this next slide sort of magnifies and reiterates the fact that this is a complicated problem, right? And we talk about where the missing plastic is, and clearly this is part of the equation of where the missing plastic is in terms of understanding how it is incorporated into the food chain, everywhere from the nanoparticles shown here or below, and then up to macroplastic. And this is an example of a ghost net that's floating around. And of course, larger top predators get entrapped and ensnared in these kinds of floating debris that's out there. So plastic in different sizes can enter the food web at many different levels. Okay, a fourth myth is only nature suffers from the negative effects of plastic soup. And this cartoon here exemplifies the fact that things like aquaculture and shipping and fishing and tourism are also impacted, as well as many other phases of our life. But I think this is especially relevant for AtlantECO, because of course our connection with aquaculture and fishing. And I think these are two aspects that AtlantECO will help to inform in terms of some of the efforts that we have in place. A fifth myth is that I'm not personally affected by the plastic soup. And this is a cartoon that came out of a workshop that I participated in last year. And it highlights the fact that, in fact, organisms from, again, the different levels of the trophic trophic food web in the ocean are accumulating different forms of microplastic. And if we consume these items, we are indeed consuming plastic along with them. And of course, this is still heavily debated as to what impacts this has on our health. And this is, in fact, a new area of study that's becoming more mainstream in different countries, including the Netherlands. And it's not only fish, of course, but other forms, bottled water and salt and a myriad of different materials now where microplastics is showing up in our food chain. And I mean, my personal interest in this connection, of course, is what role does the plastisphere play? And when we read the literature, we often see reports of higher organisms like fish being subjected to plastic and its resultant impacts with types of different organic pollutants that become attracted to it. But the role that microbes play in this transfer, for example, is very much unknown. And so there's a lot of unknowns with respect to what role the plastics and plastisphere play along our food chain. Another myth is that we really can't do anything to prevent or solve the problem of plastic soup. And to this argument, I like to present this pyramid from a GASAMP report, which is a group that was put together by the UN to help, and UNESCO to help inform these types of problems and how to deal with them on a policy level. And I really like this pyramid because it sort of emphasizes the fact that what we should be focused on is preventing and reducing the generation of waste in the first place in terms of protecting the ocean. And then a second layer, preventing and reducing litter from reaching the marine environment. And things like waste collection and improving waste management are really focused on this type of activity. And then the third layer becomes collecting litter from the marine environment, such as beach cleanups and fishing for litter, where fishermen are actually given rewards for finding derelict fishing gear, for example. And then only at the fourth level do we find things like recycling and upcycling, and then below that, energy recovery, and finally, things like landfill disposal. And I think it's important to sort of think about this pyramid and try to adapt it as best we can moving forward in terms of this problem of the plastic soup and plastic in the marine environment. But of course, the solution, which maybe we don't want to hear, is really within ourselves, right? It's very much a societal problem of solving the plastic soup problem. And I think we need to look into ourselves. And of course, we are mostly microbial to begin with. But this is something that is a message, I think, that needs to really come across loud and clear with respect to the problem of plastic pollution. Now, I do want to draw our attention to recycling and upcycling because it is part of the circular economy, which is really being highlighted now in the European Union. And to that end, I just wanted to present a data slide that I found online that talks about how we're doing in terms of being able to recycle the plastic that we do consume and use. And this is a snapshot of that that was produced in 2018. And you can see, well, I would dare to say that we're not doing that well. In the US, it's less than 9% or 9%, and the European Union writ large is 30%. And places like India are doing better. But again, this is showing just a snapshot of what's happening across the world. And of course, anytime we use a recycled plastic, we still need virgin plastic to produce a final product. And the seventh myth I want to mention is that biodegradable plastic is a solution to the plastics problem. And I think this is being put forth in various forms. And a very good example of it is with standards organizations that are trying to put together standard methods for some of the examples shown here, like recovery and recycling of plastic waste, looking at the carbon content of plastic materials, and then also biodegradation testing and disintegration standards. But in fact, I think that the reliance on things like biodegradation in the marine environment to solve our plastic problem has really kind of taken on a life of its own. And when we first started working with plastic in my group, I was attending an international conference on marine debris and presented some of our early work on members of the plastisphere and this sort of concept of microbes living on plastic, and was interviewed by a reporter from Nature News who I specifically told, I said, okay, you can say anything, but you can't say that microbes are digesting plastic. As long as you don't say that, it'll be great, because these are early days. We just really don't know what's happening. And lo and behold, of course, what does the headline say other than marine microbes are digesting plastic? And of course, this has propagated in the popular literature, and is something that, this was in June 2011. And then 8 years later, in Dutch now, we're still asking bacteria to please bacteria digest plastic. So I am certainly aware that there are definitely tools and molecules and enzymes that are capable of breaking down plastic. But I think relying on microbes alone to solve our plastic problem is not the path forward. And this is also being picked up in the media at some level. And I think hopefully this is also something that AtlantECO will be able to adopt, and a message that we'll be able to send to certainly the popular literature and the public, but also the scientific community as well. And indeed, solutions do begin with us in more ways than one. So I want to segue now and talk a little bit more about the plastisphere, as we've called it. And I mentioned that it has kind of evolved over time. And if you do a Wikipedia search, you'll actually find this definition, which states it's a term used to refer to ecosystems that have evolved to live in human-made plastic environments. This is a definition you can find online. But when we originally described the plastisphere, it was very much a microbial-centric definition. And when we started literally looking at the biofilm communities on plastic, we likened it to the biosphere, because it was this thin film of life around the plastic, as around the surface of the planet, that is really a little world in and of itself, rather. And that's because we find things like diatoms and cyanobacteria that are phototrophs and can make their own food. Things like predators such as the ciliate and its associated symbionts that live on the outside of it. And then, of course, also heterotrophs that are capable of producing or using organic compounds, and potentially even using the plastic surface itself as a source of a form of sustenance. So, of course, plastics have been around a very long time, but studying microbes that live on plastic is a newer activity. But I just want to sort of give us a little bit of a history, if you will, of the plastics timeline, starting back in the early 1900s with the production of things like bakelite. And in between then and the 50s or 60s or so, we find that a lot of our everyday fossil fuel-based plastics were invented. But it really wasn't until after World War II that the use of plastics, and particularly the production of plastics, really took off. And interestingly enough, the first publication that's often cited that describes the discovery of plastic marine debris, it happened in the 1970s. And since then, there's been a growing production of plastic, but also a growing interest in the topic of plastic marine debris. Another milestone, I think, that we're all aware of is the fact that only in 1988, perhaps, there was a prohibition via the Marpol Annex of dumping at sea. So prior to that, we were dumping our trash in the ocean, including plastic. And yeah, so this was a big event in terms of policy. Just as an aside, in the 1990s, the first biodegradable plastic was produced. And I think a really important milestone that we should all be very interested in is the fact that in 2019, the European Parliament actually started a ban on single-use plastic to be implemented over the next several years. And so this is a milestone in terms of Europe. And when we look at the literature, this estimate of 10 million metric tons per year of plastic that enters into the ocean is a statistic that was published in 2015, and the fear, of course, is that if life continues or the status quo continues, that this quantity of plastic that enters the ocean is really going to escalate to a point where it really will continue to be a big problem in our oceans. So we often hear the term microplastics, and for those of us that work on plankton, we know that there's these convenient sort of size classes that describe plankton. But one of the reasons why this topic of what size actually are microplastics comes into play is because of the definitions of my primary and secondary microplastics. So before a plastic product is produced, it's what becomes a plastic product is something called a pre-production pellet. And those pellets happen to be typically less than 5 millimeters or 5 millimeters in size. And so the original definition of microplastics took on this sort of upper size limit, I should say. And for a long time, there hadn't been or haven't been lower size limits. That's changing now within some of the standards communities, and this sort of larger size, 1 to 5 millimeter, is what's often called, now referred to as large microplastic. And then when we start to look down into the nano scale, which colleagues like Alexandra Terhal are doing, this is the zone that we're in. And again, this definition is a little bit flaky. It can either be less than 1 micron or 1,000 nanometers or 100 nanometers, depending on whose definition we're using. But suffice it to say, plastic marine debris scales all of these levels, all the way down to the nano, up to large pieces of docks and other large styrofoam and other types of plastic materials that typically end up entering the marine environment due to catastrophic events like tsunamis or hurricanes, etc. So why do we care, right? Why is this important? Well, we heard about the Ocean Health Index yesterday, and among the variables that are important in the Ocean Health Index are pollution and invasive species transport. And when it comes to plastic of any size, we have both of these happenings or these problems. And that's because plastic is considered a pollutant. So by virtue of its being, it is already part of the Ocean Health Index concern. But more importantly, invasive species transport is something that is now becoming an issue and a topic of interest with respect to plastic pollution. And that's because as early as 2003, there's a realization that harmful algal blooming algae can attach to plastic, thereby potentially being transported. Disease-causing bacteria like Vibrios are known to associate with plastic as well, both in coastal regions, in sediments, and in open ocean environments. And finally, something maybe that's less obvious is the displacement of native species in terms of things like ghost nets. And the particular example that I like to give in this regard is we have encountered the occurrence of things, endemic species to Sargassum, which is an open ocean floating macroalga that actually had, so we encountered a ghost net that had endemic species that are normally found on this Sargassum macroalga that we're actually now associating with this artificial ghost net habitat. So not only are we thinking or looking at the displacement of microbiomes of organisms that are associated with plastic, but also native species that are becoming displaced by this new habitat that we are forming. Now, this topic, so I'm a microbial ecologist, and it's not very common that people get excited about microbes. But when you add plastic to the equation, it turns out that people become very fascinated with the topic. And in some ways, it has been a wonderful mechanism to educate people about the importance of microbes and microbial ecology. So there's a huge science component, of course, that is related to the topic of the Plastisphere, but it has really served as a tremendous education and outreach outlet as well. And I'm very, very pleased and excited to know that AtlantECO is going to be focused on this sort of segue, if you will, on education and outreach for plastics and other aspects of AtlantECO as well. In addition to education and outreach, policy has a really important role with respect to plastics and the plastisphere. And I have actually personally served on GASAMP, as I know others have in the AtlantECO group as well. And I think it's really important to provide this kind of service to the community and to the policy groups, because they really need our expertise to help inform decision making. And in some cases, some of the regulation that's happening now, especially in the EU, is going ahead of a lot of the science. And that, to me, is a bit troublesome. But hopefully some of what we'll be accomplishing and achieving in AtlantECO will help inform policy in a serious way. And finally, I do want to mention industry, because industry is also very invested in the topic of plastic marine debris and in plastic debris in general. And in particular, now with extended producer responsibilities, they're especially sensitive about how their products are affecting the environment. And again, there is a role we can play in joining these organizations and helping provide our expert opinion on certain aspects of the science behind standards science. So with that, I want to segue now into diversity, function, and fate aspects of the plastisphere. And some of the main questions that my lab and other labs that ponder this field include the following. Is there a core plastisphere microbiome? Are plastisphere communities phylogenetically diverse but functionally equivalent? And finally, what is the ecological role and impact these microbial reefs, as they're sometimes called, have had on marine ecosystems? And to this end, we use a variety of methods to study the plastisphere. And these range from looking at the types of plastic that are out there and in addition to what microbes are living on them. And there are now sophisticated micro-Raman and micro-FTIR spectroscopy that help us identify what these microplastics are based on reference spectra. In addition, we like to visualize the spatial structure of the plastisphere by using tools like scanning electron microscopy. And this is an image of such a snapshot of that community. And it can tell us something about perhaps which microbes are interacting with each other. And of course, that's of interest to Working Group 5 and understanding the interactome. And certainly there's a lot of interactions that we can already visualize, and hopefully molecular methods that help us connect these different interactions will help us go even further beyond that. And of course, there are culture-independent methods that have been widely used now by several groups, including Illumina and MinION next-generation sequencing. And this is a way, of course, of probing more than just who's there, but what they're doing and in terms of their entire genomic structure. And then finally, culture isolation is still a traditional but powerful way of looking at different community members that are present in the plastisphere. But studying the plastisphere, turns out, is quite complex in that there's a lot happening on a single piece of microplastic, particularly out in the open waters in these ocean gyres. And here I have that petri dish that has the microplastic in it. And I like to say that every piece of microplastic really has its own history, because even within a single net toe, any one piece of plastic didn't necessarily come from the same location. And it also didn't fall in the ocean at the same time. And so this issue of time is really important when we think about the communities attached to plastic and even the plastic itself. And that's because there are different forces, of course, that act on microplastic. We have photodegradation and chemical degradation that are acting to change the surface of microplastic and break it down in some cases. And also on top of that, then we have biology. And biology usually tends to really obscure, in particular, the community structure that's present there. And what I mean by that is that when we first started studying the plastisphere, there was this argument, well, you don't have replicates of your samples. And so this is an example of a replicate from a single net toe. And you can see replication doesn't really make sense in terms of studying this community, because every piece of microplastic has a different history. Now, of course, occasionally you can get the same piece of microplastic in the same netto, but if you saw Eric Van Sevilla's some of his models and where a particle ends up, depending on where it starts, you can understand that the problem is complicated in terms of understanding and getting a general picture of what lives in the plastisphere. So we use a battery of different tools to study the chemical structure. Of course, I've already mentioned Raman and FTIR, but also SEM, and something I'll mention in a little bit, classy fish also help to understand some of the chemical and photodegradation kinds of phenomena that can be attributed to microplastic characterization. But if you're looking at the who, where, and what, then you're turning mostly to omic type technologies such as amplicon sequencing, which is looking at just a single portion of a marker gene and understanding the community structure. And each one of these different bands here represents a different group of bacteria, or OTU, operational taxonomic unit, within a community. But then, of course, we also like to visualize. So SEM and fluorescence in situ hybridization also get at who's there and can often tell us something about their spatial orientations. And newer techniques such as stable isotope probing are also being used to help clarify who's doing what within a given community as well. So I like to say that plastics exhibit a highly variable community structure when compared to seawater, regardless of the location. And just to bring home that principle, what I have here are the communities. So each color represents a different bacterial OTU. And you can see that if we look at these 5 pieces of plastic, 5 pieces of plastic from the same netto versus the surrounding seawater from 2 distinct ocean basins, it's remarkable in the structure that you see, with the Atlantic and the Pacific water communities being more similar to each other than either are to the plastic communities. And so this was a study based on just the V6 variable region of the small subunit ribosomal RNA. But this is a pattern that we see repeatedly in not only our studies, but those of others when they look at different parts. In this case, this was the North Atlantic. These are pieces of plastic from the North Atlantic subtropical gyre. Another point of discovery and fact that we know about the plastisphere is geography appears to matter. And this we discovered when we compared communities in the Pacific and the Atlantic basins based on 2 transects. And I want to add that these studies that I'm mentioning now were actually done on individual pieces of microplastic. So not pooled samples, but individual pieces. So each one of these, this is a biplot. So this is a multi-dimensional analysis, multivariate dimensional analysis. And what it does is it tells us something about the community structure within a given sample. So each one of these, in this case, triangles or circles, represents the composition or beta diversity within a given community that we analyzed. And what you can see right away is that there was a separation between the Atlantic and the Pacific communities in terms of their community structure, such that the Atlantic communities were more similar to each other versus the Pacific. And this seemed to make more of a difference than the actual polymer type, as you can see in the right-hand side. And of course, I will argue that the sort of distribution of different polymers is not what we would like it to be in terms of the total numbers of different types of polymers. But in fact, this is nice and convenient because what it does is it actually shows you in a typical analysis the contribution of different polymers that you see, at least floating on the surface. And what we tend to see is mostly polyethylene, followed by polypropylene and polystyrene in our expanded polystyrene in this case in our sampling and in our data sets. So, excuse me. So, a convenient way to study the membership of the community membership of the plastisphere is not to actually collect samples from the environment, but instead to put out baits, if you will, and incubate plastic in a given environment, and then look over time to see how the community changes. And this is an example that we recently published in a review, and it shows sort of the succession, as you would see in a forest, let's say, after a fire, of what colonizes over time. And so we can combine image data and we can combine molecular data to look at the evolution of the community over time. And one of the repeated patterns that we see in these kinds of studies is that things like diatoms, and in this case, this was revealed by their chloroplast DNA, are definitely among the earlier colonizers that we see. And we see this pattern repeatedly, and then they decrease over time and they're replaced by various other types of organisms, but especially different bacterial groups come and go as the community matures. So this is a powerful way of connecting image data with also a microbial data. And again, I know that this is something we want to do in AtlantECO, and I think it's a really nice example of how we can do that. This is this SEM micrograph demonstrates, I think, an important point about the plastisphere, and that what you're looking at here is a diatom frustule, and it's the shell of a dead diatom, but what you see inside of it are these bacteria. And so I think a lot of times when we think about biofilms, we tend to be biased towards bacteria and archaea. But in fact, this is, I think, a prime example of the fact that microbial eukaryotes, in particular diatoms, can also form biofilms on the outer surface of plastic. In fact, they are very important in early establishment of biofilms in the plastisphere. So I also want to mention that when you do a survey of the literature, and there are growing numbers of these data sets now on incubation experiments that have taken place in different parts of the world, we see that a majority of these experiments that are indicated in blue are incubation experiments. And these tend to be the dominating kinds of data sets available. And I think what AtlantECO will bring to our knowledge is more information about actual individual communities on naturally occurring plastic marine debris, which I think is wonderful. Now, in addition, my own group, we've been exploring the literature as well, and through something called VAMPS, which was the original data portal for those of you who remember ICOM, which was the first census of marine microbes in the ocean. This was a database established for this particular project that was funded through the Sloan Foundation, and I helped manage at that time. And what we've done here is to harvest data sets from the literature using different variable regions. So the small subunit-ribosomal RNA marker gene has these different variable regions that various groups have used to do amplicon sequencing and look at microbial profiling across different types of samples. And if you survey the literature, you'll see there's all these different variable regions that have been used. And so what we've tried to do in the VAMPS Plastisphere portal is harvest all this data and make it available to look at comparatively using taxonomy-based, not OTU-based analyses. And so we've already kind of gotten a jump start, hopefully, in terms of being able to ingest some of this data into AtlantECO and some of the efforts there. Now, you might say, well, okay, that's okay, but it's taxonomy-based and the resolution isn't fantastic. And it's true, it's not the best. It's not as good as looking at individual regions and comparing them directly on a sequence-based level. But one of the things we were able to do using this more blunt approach was to compare, at the time when Caroline and I got together to work on this review, was we were able to compare her samples from the North Sea with our samples from the coastal Atlantic, open ocean Atlantic, and the open ocean Pacific. And one of the nice things that this did was it helped us develop a sense of higher order, so looking at phyla and classes, some of the major components, some of the major members of the plastisphere. And what we were able to do after we had done this exercise is we were able to produce group-specific probes and use them in a new method that we've recently published on called combinatorial labeling and spectral imaging fluorescence in situ hybridization. And this is a method that kind of goes beyond the scanning electron microscopy method, because not only does it let us visualize individual microbes and look at their proximity to each other in the biofilm, but it lets us now understand who is who at least at some level of taxonomy. And in theory, well, theoretically, you can use this method to simultaneously visualize hundreds of microbes at one time. Practically speaking, it works well with about 15, but again, you can go beyond that. And in our study, we did that in particular to look at the communities that develop over time, particularly over the early colonization timeframe. And the reason we think this is important is because this is the time point where microbes are literally directly interacting with the plastic itself. And if there's anything to be said in terms of understanding the influence of the polymer on the developing communities, kind of this is where we think it's important and interesting to examine. And what you'll see, these are the different colors of the bacteria and the microbes represent these different colors in these major groups. And the larger white cells are diatoms. And again, this is an early colonization, so we tend to see these early on in the development of the biofilms. And what you can see is there's really quite a bit of variation that we have in the North Atlantic versus the tropical Atlantic versus the Bodensee, which is right off of NIOZ in our dock there. And admittedly, these were done at different times of the year, so temperature clearly was influential in affecting or in shaping the underlying communities. So that's great. So we can learn about sort of who's there. But of course, many of us are very interested in their genomic composition and what we can learn about the metabolism. And again, this I know is front and center in terms of AtlantECO. And so in my own lab, we've been taking a combined approach of culturing, as I mentioned before, but also metagenomics in terms of reassembling the entire genomes of given Plastisphere members. And one of the challenges in doing that is that in many of the Tara Ocean collections, there was the luxury of being able to size select different communities. And of course, this has the advantage of being able to simplify the resulting communities. But in biofilms, you're up against a different issue, which is not being able to really remove well, not only microbial eukaryotes, but also microscopic animals in many cases. And so what we're left with is this issue of dominance in many cases of eukaryotic organisms. And this makes a challenge for trying to subsequently reassemble genomes. Nevertheless, it is possible to do comparative genomics and comparative metagenomics, as I'm showing you here. This is a diagram that's rendered through a platform called Amvio. And all of these outer colors here represent metagenomic bins, where we think we have parts of genomes or metagenome assembled genomes that represent commonalities between some of these different samples that were co-assembled and then subsequently mapped with individual reads. But I don't want to get into the details of this. Suffice to say that we do find members, particularly of biofilm communities, that are common across some of these kinds of samples. And things like Ultramonas and Erythrobacter and even diatoms are sometimes found across multiple samples. And alongside these particular genomic bins, we see evidence for the types of genes involved with biofilm formation, which of course makes a whole lot of sense. So at least, despite the diversity we see in the community structure, at least we can see a semblance of commonality between some of the members that at least are associated with the biofilm. And going beyond that, we can then of course try to assemble those genomes, and this is an example of different bins using a method called ESOMs that were produced as part of an analysis of one of the metagenomes that I showed you earlier. And in some cases, we can get fairly high, what appears to be fairly high recovery of some of these MAGs. So then we can take these genomes and compare them, of course, to something that might be found in the water column or genomes from other similarly assembled metagenomes. So the last sort of topic I want to cover in my talk here, in my lecture, is this concept of degradation versus biodegradation. And you know, if you look at the literature now and do a search for biodegradation, you really come up with a lot of different manuscripts and papers. And sometimes they actually explicitly mention biodegradation, or they're in a topical area under biodegradation. But I think a lot of these papers, instead of looking at real biodegradation, what they're looking at is in fact degradation of plastic by enzymes, etc. And the reason for that is because the sort of concept of biodegradation as a mechanism is actually fairly well studied in terms of what happens, at least in terrestrial environments. And this is a schematic from a review that was published in 2008, and it depicts a microorganism and its interaction with a plastic surface that ultimately leads to CO2 and water and biomass effectively. And so these are external enzymes that get secreted and then act upon the surface of the plastic. And this is most commonly effective in settings where you have things like polyesters or hydrolyzable types of plastics. And we know there are things like lactase enzymes and more recently petases that are involved in this kind of microbial transformation of polymers. But in fact, when I'm asked if microbes are biodegrading plastic in the ocean, I have to pause and say, well, I mean, I think when you're thinking about the biodegradation of materials in the ocean, you can't only stop at the polymer fragments. You have to go all the way down to CO2 and water and cell biomass. And I think what often escapes a lot of publications is this issue of time. And when we're talking about biodegradation in any environment, we need to put a timestamp on it, because we don't want biodegradation to happen on the timescales of years to decades to centuries. We want it to be within days to months to weeks. And so I think it's important to understand this, and it's important to understand that the definition of biodegradation is one that involves complete biodegradation or microbial assimilation of the plastic substrate as measured by CO2 evolution or CO2 and methane, depending on whether it's aerobic or anaerobic. And in the marine environment, there's a lot of issues that surround this, including the fact that there tend to be fewer microbes in certain parts of the water column, let's say, versus things like sediments and certainly soils. There's high hydrostatic pressure, which may or may not come into play. Certainly cold temperatures, I think, are was one of the biggest impediments to biodegradation in the marine environment. And then also sparse nutrients, depending on where you are in the water column. So one of the solutions that have been put forth for dealing with marine pollution, as I mentioned in among my myths, was bioplastics or biodegradable plastics. And I really like this graphic here that is available through European Bioplastics, because it does a really nice job at explaining some of the confusion, or elucidating some of the confusion that can happen with respect to certain terms regarding biodegradation, biodegradability, bioplastics, and bio whatever. So what you see here is a biplot, and the x-axis represents degradable, non-biodegradable versus biodegradable, versus the y-axis, which is feedstock, or whether it's bio-based or fossil-based. Okay, so the y-axis represents the beginning of life, and the x-axis represents the end of life. And why this is important is because when you come across the term bioplastics, as you can see from this biplot, bioplastics can be non-biodegradable and bio-based. They can be bio-based and biodegradable, or they could be fossil-based and biodegradable. So simply using the word bioplastics isn't meaningful. What is important is the fact that a bio-based material comes from a feedstock. And unfortunately, the terms biodegradable have kind of entered the sort of public arena in terms of two materials that are often referred to either as oxo-degradable or oxo-biodegradable. And as the name implies, they sound like they may be good for the environment, but in fact, these materials have been banned by the European Union. Now, that's great for the European Union, but unfortunately, these kinds of materials that are effectively described as biodegradable are still being sold in North America and South America and also Africa. So what my lab has been very involved with is starting to look at biodegradation from a microbial perspective in terms of genomics. And as I mentioned, biodegradation is measured in terms of CO2 evolution. And so ideally what we have is we have the evolution of CO2 and biodegradation rates that follow some kind of standard. And these standards exist, but in fact, the one that has been adopted by many groups but has recently been withdrawn is one from the ASTM, International Organization, Standards Organization, that requires 30% degradation within 180 days versus any other rate. But what you can see here in red is a PHA polymer, which is a biodegradable polymer, versus something like a paper bag, which actually has a slower rate of biodegradation. Now, my lab group, and in particular my graduate student, Fons de Vogel, we've been looking at this original consortium that was defined by the standard and trying to better understand the enzymes involved in the consortium and how they might be contributing to biodegradation. And we've been using simple tests like this, which is called a clearing test, that helps help us sort of screen the initial behavior of some of these microbes. And I imagine that these are the kinds of tests that we might be able to also adopt in AtlantECO. Okay, so I'm rapidly running out of time, so I want to leave you with some conclusions and outlook. So the big conclusions and the big take-home messages are the following. First of all, microbial populations that colonize plastic marine debris do it really quickly, and they can be dominated by diatoms even within 1 week. And so it's not only bacteria that are colonizing and contributing to the plastisphere. What we've discovered so far, and we've sort of touched the tip of the iceberg, and that is that very few taxa appear to be members of shared core microbiomes, but biofilm formers are among them, perhaps not surprisingly. Thirdly, biofilm formers may include genera of bacteria or eukaryotes, so they're not restricted to bacteria. And things like comparative genomics, both of cultured isolates, but then also MAGs, can help tell us something beyond who's there and possibly what they're doing, and give us a window into their functional potential of the underlying communities. And finally, since I started with sort of take-home messages and myths, I want to leave you with some take-home messages regarding plastic marine debris and biodegradation. So first, and I think a really important message, is in fact that despite the fact that we do have ocean basins and we do have seas, many seas, I like to think that there is one ocean, because what that implies is that it's shared by everyone. And what that means is that we all have to be stewards of our ocean. Marine debris in particular does not respect EEZs. And so this is a really important point that needs to be reiterated and really extended, especially to the next generation of citizens of our planet. Secondly, out of sight shouldn't be out of mind when it comes to microplastics. And unfortunately, some of the early types of biodegradable plastics that are out there are really not meant to biodegrade. They're meant to disintegrate, and then they form microplastics. And so we need a good way of understanding the fate of plastics in the ocean, and getting at this question of what that looks like is a really important aspect of what I think the future of plastic marine debris research needs to focus on. Thirdly, reduce, reuse, and recycle. This is maybe trivial and trite at this point, but I think it still is very much an important philosophy for all of us to follow. Fourthly, biodegradable compostable plastic is a solution to the plastic problem, but not the solution to the plastic problem. And I think there's a lot more research that needs to be done to help us all feel comfortable with what its role is with respect to the marine environment, and what kinds of materials are safe to use in the marine environment and which are not. And the reason for that is because we need proper waste infrastructure and composting facilities in order to make some of these newer materials reliable and proper replacements for conventional plastic. But beyond that, we also need to just improve our waste infrastructure at large, and especially our recycling capacity, because as we see, we're not doing as good a job as we should be with respect to recycling opportunities. And importantly, marine environments are not composting facilities. They're not a place where anyone should ever throw their litter intentionally. And And this is important. It's a huge message, because it's part of the reason why in some, at least in the United States, in some states, the word biodegradability is not allowed to be even placed on a product. And that's because of the fear that this will be a license to litter. And the last thing we want is to allow the public to take on this societal behavior. And this has really been a huge issue, and one that might even prevent us from coming up with more sustainable solutions to our plastic pollution problem. And finally, and perhaps most importantly, global problems require global cooperation and collaborative efforts across international standards bodies. And this is also challenging, because all of us have different agendas, and even standards organizations have different agendas. And in particular, they're in place to make money. So while they could be coming together to talk about the best and most effective ways of creating standards that are realistic and reflective of what's happening in the marine environment, they don't. And this I also see as another barrier, and perhaps even one that AtlantECO might be able to tackle as well. So with that, I leave you with my thanks, and I just want to acknowledge various funding agencies on both sides of the Atlantic that have helped fund our research, and also all the collaborators that have been with my group throughout the years, helping to contribute to what we know my group has at least contributed so far to the Plastisphere. Thank you.
So thank you, Linda. It was an amazing talk, another amazing talk, I would say. But and in addition, I think this confirmed that let's say the structure of AtlantECO is really actually good in terms of trying to address these issues. I mean, we cannot separate the microbiome, the understanding of the function in the microbiome from the problem, the plastics. And I think it's also nice to see that tools are like genomics, genetics are also the same, imaging. And so, yeah, this comforts me a lot, I have to say, and it will be interesting indeed to see what is in the water at the same time, and to try also to understand, for instance, the contact rates. No? So, try to see where it is coming, where the stuff is coming from, how it does live in the free living form. Also, I think as a way also to model the growth of the biofilms. So that was really great, but I don't want to take more time, and I think it's better to take questions now.
Okay. Yeah.
We have a question from David Jones.
Yes, hi. Thank you, Linda. That was an excellent talk. My name is David Johns from the MBA in Plymouth, and it was just a quick question really about what your views were on some of the contamination issues with microplastic work. You know, you showed some pictures at the beginning of the some of the methods such as manta trawls, and I know in the UK, if you're talking about policy work, people are very reluctant to use sort of samples taken from underway to report on microplastics, and instead they're using sediment records and fulma, like a seabird stomach content, because there is this concern about contamination. I just wondered, sort of all the data sets you've used, if you've come across that issue or you think it's a major issue.
So by contamination, you mean plastics from other sources and
Sure.
Yeah. Well, for the floating, like larger, what I call larger than 350 micron size microplastics, at least if you're out in the open ocean, we don't worry about that. We don't worry about contaminating those kinds of plastics with those kinds of materials. But when it becomes an issue is when you start to look at the smaller size fractions, indeed. And this is definitely a problem and requires another level of care. And yeah, it's challenging. You know, it's really challenging going beyond starting to get at the micron and submicron level of work. Not only in terms of the kinds of equipment that you need that are required to analyze and characterize those smaller size fractions, but yeah, just in terms of knowing, you know, there's another layer of controls that need to be added at the same time. So it's definitely challenging to go to the lower levels of study for sure.
Oh, great, thank you.
Yeah. Oh, should I read these?
It's, yeah, question from Silvia Sines.
Yeah.
Oh, okay.
Hi, Linda. Very nice to see you, and always your talk very, very good. Great, as always. I have just a curiosity about. You mentioned that you also have cultures, right, from the plastic that you build also the beans, the metagenomics beans, and some of your beans are really at taxonomical level, but similar, very similar to very well-known isolates like Erythrobacter and some other. Did you find any perfect match between the isolates and the max at some level? Did you have compare?
Sorry, did I compare the known cultures versus the ones from
the culture? Yeah,
culture.
From the belongs, for instance. I'm sure that you will get a lot of the Erythrobacter isolates for sure, and you have also the beans from Erythrobacter. I don't know, you try to compare.
We're doing that now. Yeah, it's taking time. I mean, I just moved my lab 3 years ago, and so I'm sort of coming up to speed with that, and it was sort of right in the middle when we were gathering a lot of these data. But yes, the nice thing is we do in fact have a lot of the isolates, like we have cultures of a lot of the isolates that in particular tend to be the biofilm forming parts of the community. And so, so yes, that is something that we're actively doing now and that we hope to be able to learn from. So that's part of what we're trying to do now. Yeah.
Thanks.
Yeah. Thank you for your question. Jean. Jeff, yes.
I have a question from Jeff.
Okay, sorry. In the Netherlands, yeah, but I know I'm not sure what you are in France.
Hello, Linda. It was really nice talk. I really enjoyed it. As far as we understand, we have a really specific diversity in plastic compared to the free living or particle attached bacteria. We know that you present that in each plastic pieces, you can have really different communities, but we have general tendencies that say that we have kind of core microbiome and core plastisphere, we will say. So let's try to find this, and this is one of the aim of the AtlantECO project. And we also see some biogeography, relation between Atlantic, Pacific. We also found some the similar data with the Mediterranean Sea compared to Pacific, for example.
Yeah.
So this means that probably the environment matters, and this theory about everything is everywhere, but the environment selects. One of my questions will be, I would be really interested if you thought about this, and if I think it's in AtlantECO, it would be really nice to look at this, meaning how much the environment selects the free living bacteria versus the particle attached bacteria versus the plastisphere. And since we will get a really rough idea about the diversity in the Atlantic because of this project, I think it may be really interesting in this aspect.
Yeah, no, I think it's fantastic. Yeah, I mean, even just the Atlantic, right? Being able to compare the North Atlantic and the South Atlantic to the tropical Atlantic, etc. Yeah, I agree that so what at least seems to be happening from our estimations and really those of others is that the early stages of colonization are influenced by polymers type. And so how quickly biology takes over is, I guess, part of the question, and whether that's faster in the tropical regions, let's say, or slower, or yeah, I mean, I think this is a really interesting question, and one that we could probably also tackle in terms of experimental types of setups as well. But yeah, I agree that it's fascinating. But of course, we talk about plastic as this monolithic entity, right? Even polyethylene isn't just polyethylene, right? There's all this myriad of additives. So this layer of complexity somehow needs to be, deconvoluted. And we need to better understand really what the impacts of not only the plastic, but everything that's added to it, also how that influences the resulting communities as well. So, but yeah, I'm excited. I think this is really going to be a great opportunity to really delve into some of these questions. And learn more about, yeah, what is really shaping the communities on these pieces of microplastic. Yeah, but thank you for your question. Eric.
Yeah, Linda, I have a question too. Fantastic talk. I really enjoyed it. Very nice overview. So I want to pick your brain on this Coy-Koolmans theory that I also briefly mentioned yesterday about the yo-yoing of plastic between the surface and then at depths, where the biofilm would grow, it would decrease in buoyancy, particle would sink, biofilm would be removed and would go back up.
Yeah.
Is it something that you think is possible in the real ocean? And
Yeah.
if so, how would we measure it?
Yeah, it is possible. We've actually seen it with our own experiments that I didn't talk about today. How to measure it is a great question and a great challenge. Yeah, I mean, I think it needs to be somehow informed by laboratory studies, but I think those are possible. Yeah, the way to do it is to really have very controlled pieces of plastic, and where you really have very precise thicknesses so that you can really be sure that you're comparing apples and apples and oranges and oranges. That's part of it. But yes, just to give you some insights into that, you know, we think about and we focus on smaller size pieces of plastic, but at some point, for example, micro animals will attach to plastic. And some of these animals have ballast of some form, whether it be a skeleton or a shell, and that can weigh down the plastic. And of course, once those organisms leave or get detached in some way, then that piece of plastic can become buoyant again. Or another very interesting phenomenon, if that piece of plastic disintegrates or breaks, pieces of it can then become buoyant. So there are different ways in which the density of a given piece of plastic can change. And so those are things that, yeah, all have to be taken into consideration. But I don't think they're beyond the realm of at least coming up with reasonable parameters to estimate, but I think it requires laboratory experiments. So
I would like to add quickly, because we will discuss this in a few minutes in the next session, which is on the activities, but I think it would be interesting maybe to look at in the sediment traps to see if we can have evidence of change in the community after the settling or during the sinking. Because
yes,
indeed it's a very important, so if there is actual respiration of the predators actually on these bacteria or phages, I don't know, but we'll talk in a minute. But I think yeah, we can get back to that, because there is one specific activity in the project that could help to address this.
yes, I agree completely. The sediment traps I think you're referring to, right? Yeah, I think that's a nice way of looking at that question. I mean, we know there are non-floating, so polyethylene, high density polyethylene, low density polyethylene, we know we find them at depth, and so they're getting there. And they shouldn't be, because they shouldn't be. They're lighter than seawater, but less dense than seawater. But they are, and we know that they're being, something about them is being, their density is changing, and biofilm formation is definitely part of that process. Okay. Yeah. Shall I
Let's
stop?
say two more questions, one by one. Sorry, I cannot see the surname.
Sorry, did I do something? Oh.
No, there are two more questions. One by Yvonne.
Fabio Benedetti.
Hi.
Oh, hello.
Can you hear me?
Yes.
Oh, okay, interesting.
I can only see your eyes and your nose. There we go. Okay.
Yeah, my question has to do with DNA extraction from the plastics. So why I've read most of your work, but I think the one you presented that had to do with the evolution of CO2 and trying to measure that.
Yes.
Your lab group used incubated plastics, right?
Yes, those experiments are done in the lab. That's correct. Yep.
Okay, and those weren't natural sourced plastics from the marine environment. They were like commercially sourced.
No, those experiments are done with very controlled, known composition of polymers, and they're done in the laboratory.
Yeah, but were they sourced from the natural environment, or like
No, they were not sourced from the natural environment. They're known composition of polymers, and they're in this case they're biodegradable polymers, and so we're looking at the rate of biodegradation of those biodegradable polymers.
Okay, so my question is, what challenges were there any challenges in terms of extracting DNA from those incubated samples and from the natural plastics?
Oh, in general, there are challenges associated with extracting the DNA from particularly environmental plastics, not so much incubated plastics, because you can have larger pieces if you need more biomass. But in general, working with microplastics, the biofilm varies in thickness and in extent. And so it is challenging at times to extract DNA from individual pieces. Of course, you can always pool pieces and get more that way. And at some level, I think, yeah, it's necessary to do this depending on what you're going after. But we have also developed low biomass protocols so that we can not do that. Does that answer your question?
Yeah, sort of. But would you like care to share what kind of kits was used? Is it like a soy kit?
Yeah, no. Our protocols are published. I can send you some papers and maybe a more detailed protocol. But we actually use a commercially available kit. Although in terms of some of the different habitats, it's probably a good idea to think about adapting those kits. We use a kit called PureGene. It's now owned by Kyogen, and we've been using it for a long time simply because the buffer can stay at room temperature. So we often ship samples, and we worry about compromising our samples if they're lost or left at room temperature. So we just use a buffer that stabilizes the material so that the DNA won't be compromised otherwise. Yeah, but I'm happy to share those protocols with you if that's something that you're interested in trying.
Yeah, I am. Thank you.
Welcome. Thank you for your question.
So one last question from Fabio.
Fabio, okay.
Yes. Yeah. Hi, Linda. Fabio, so from Zurich. Thanks again, of course, for your talk. I was just wondering if there was evidence of larger organisms such as microzooplankton or even larger zooplankton, if there were evidences of this kind of stuff actively detecting and grazing the microbial plastisphere. But I'm not sure whether the current approaches used by the microplastics community actually are dedicated to such observations. Because I know some of them do that with, for instance, organic matter aggregates. And I was just wondering if they could also do that with microplastics. Thanks.
Yeah, thank you. Right, so the challenge always with microscopic animals is visualizing them, maybe to confirm that they are doing something in the community. Because of course there's this issue of eDNA, right? The plastisphere can be sticky. And so you always have to be a little bit mindful that you may be picking up eDNA when you're studying the plastisphere. But I think some of these network analyses perhaps will start to unravel whether there are these relationships, and I think that could be a fruitful approach. We do encounter metazoa, particularly larval forms of metazoa, microplastics, but at some level your microzooplankton are too large to actually be part of at least the plastisphere in the lower size limits of what we have been working with. When you start to get to macroplastic, then you get a very different view of what's going on. But at some point, a piece of plastic is too small to be colonized by some of these larger microzooplankton, if that makes sense.
Yeah, okay. And also meant not necessarily colonizing it, but just viewing it as a particle full of food. So just hunting towards it, grazing it, and then leaving the environment of the.
Yes, for sure. Yes, people have found that they do get ingested. Yeah, and I think, you know, looking in the gyres for that, I think is something, I mean, I feel like it's been done, but maybe someone can correct me. Yeah, I mean, we know that zooplankton will consume the plastics, and at least barnacles for sure, and there are other size classes that do as well. You know, the diatoms in particular produce infochemicals that a lot of organisms chew onto as food, and so they definitely would attract zooplankton of larger size classes, without a doubt. So yeah, I'm just trying to figure out how you would study that in the wild per se, other than looking for it within the guts of the predator. Yeah.
If I can chip in,
Yeah.
I remember a paper from a few years ago by Julia Reiser, who investigated indeed piece of plastic in the North Atlantic gyre, and she found scraping by marine worms, for instance. I think I remember. And an important point about that, I think that we also often forget, is that the larger zooplankton, as you just suggest, Fabio, they can also play a role in fragmentation. So how do you come? I mean, one of the big questions that's also still open is how do you get from a very large millimeter-sized plastic to a very small nanometer-sized plastic? We don't really know what that pathway is, and grazing, scraping, ingestion, egestion, I think, can also all be part of that process.
Yeah. No, that's a good point. Thank you, Art, for pointing that out.